Okay, I think we should get started. So take it away, Jane.
Okay. I'll try not to make this too long, but hope some of it will be interesting. So the idea is that validation shouldn't just be a check at the end. It's something you should do early on, as Pavel also said. And the main point of it is we hope to have validation that will show you how to correct problems. And that works very well for model validation and map to model or model to map validation up to about 2.5 angstroms. But we're now, the whole community is really struggling with how to do validation at 3 to 4 angstrom resolution, where all of our earlier validations are being restrained. So going to sort of take you through a quick tour of the earlier validations and what parts of them really are still good, and then show our Multi-residue validation that's in Phenix and some other things in Phenix and elsewhere that now is beginning to make things work at 3 to 4 angstroms better. So I've got to figure out now what, yeah, all right. So the general idea is that at least if your data is reasonable, then even at very low resolution, cryo-EM is showing us just amazing things, surprising, neat things. And one of my favorite examples are these long helices that form on each side of the growing genome of the SARS virus that stabilize their very long genome. And we had no idea that it worked this way until this particular cryo-EM structure. And so that kind of thing doesn't depend very much on local accuracy really being that great. We learn all sorts of things even so. But if you want to do certainly things like drug design, but even understanding the biology at an active site, you need much closer, more accurate local information. And so validation helps with that, particularly when it can show you how to correct problems. And on our lab's validation, so I'll often talk about it as Multi-residue, but essentially all of it is in Phenix at this point and in a number of other programs as well, although not as thoroughly. We looked at what got us started on this was all atom contacts. We were doing protein design and we realized that we needed to have much better accurate evaluation of how atoms were actually touching each other, including the hydrogens. And that got us started. Then we had to update other kinds of validation to go along with it. And more recently, we have added things like cis-non-proline and kablam evaluation and a number of other things that I'll talk about at least briefly, and RNA validation. We're one of the few validation sites that actually pays any attention to nucleic acids. And DNA we find is extremely hard to validate for reasons I can explain if anybody wants to know, but we've done pretty well with RNA. And so obviously Ramachandran is one of the real classics, and we updated it a while ago with a top 8,000 database with about 1 million residues and divided it into 6 categories. So the general category is what you're used to looking at. And then we added isoleucine and valine, which are really very different. You can see how different this picture is from the one on the left. And not just that it doesn't like the right-hand side of the Ramachandran plot. It has different shapes and preferences even on the left side. But I want to mainly, at this point, try to explain all atom contacts. So the sort of picture that one is used to looking at and think of as full detail is actually missing half of the atoms, which are the hydrogens. And if you're going to leave out half the atoms, that's the half to leave out, I must say. But they are the twigs on the outside of everything. And so most contacts include a hydrogen on at least one side and often both sides. And that includes contacts inside of a molecule that help it fold up. And it includes contacts to ligands and contacts to other chains and nucleic acids and whatever else. If you're really going to look at specific contacts, you have to have the hydrogens or you're not getting the right answer. And so the way we do that is that we put a ball of dots on each van der Waals surface of all the atoms, including the hydrogens. And then basically we roll a little ball. It's only 1/4 angstrom in radius. So we're looking at pretty tight contacts. And when it doesn't touch anything else, we don't do anything. But if it touches another non-covalently bonded atom, then we lay down a dot. And if it's far apart, if it's near to 1/2 angstrom, it's blue. If it's very close to van der Waals contact, it's green. If it overlaps very slightly, that's yellow. None of this is accurate enough to look at 1 or 2/10 of an angstrom. Atoms are not really spherical. Our numbers are not perfect, let alone the structures are not perfect. So we don't pay too much attention until we get to 0.4 angstrom overlap. And then we start showing hot pink spikes that we call clashes. And we hope that people would find those uncomfortable and want to get rid of them. I think we probably did that too thoroughly. You're always going to have a few left, maybe, that you can't explain away. In general, 0 is not the goal in outliers. The clashes are a little more absolute, but most outliers are done by statistics in very good structures. And the very good structures have small numbers of these outliers. So, 0 means that in one sense you've overfit it. That's not necessarily a bad thing at low resolution, because as Pavel made very clear, you really need a lot more extra information in order to make refinement and model building and refinement work at those resolutions. But don't think that you have to get rid of every one of them. What you want to do is get rid of most of them so you can find the ones that are actually functionally important and interesting. And when you have real genuine outliers, they're apt to be interesting, that they wouldn't have been conserved in the structure if they weren't useful for something. So the reason we believe this is that indeed all atom contacts look marvelous in the good parts of high resolution structures. And you discover when you start paying attention to them that you can really improve things. So here's a histidine, which happens to be at an active site. And in the original structure, it had a clash to this water. Well, okay, so that's not such a big deal, but it has a clash to a backbone carbonyl oxygen, which is an obligate acceptor, and yet it's a CH instead of an NH that's pointing at that atom. And if you flip over the histidine, then you get hydrogen bonds, really nice hydrogen bonds to both, in both directions. So just a quick run through. I was talking about clashes, H bonds, and van der Waals, all atom contacts. And we also have bond length and angle markup, and C beta deviations are a more complicated kind of multiple angle single parameter. And cis non-prolines. Cis prolines are fine, but cis non-prolines are not usually. And you know, rotamers and Ramachandran, and then ribose pucker is important for RNA. And then we'll be talking more about kablam later, which is a multi-residue substitute for Ramachandran. It can show problems when Ramachandran has been refined away. And people using this have really improved structures. Here's an initial structure with a bunch of clashes and rotamer outliers, and particularly this N-terminal end here is really in trouble. After some fix-up, that turned out to be a switch between the side chain and the backbone right back there. And here, afterward, there are only a couple of tiny clashes, and there's a new sulfate. And it's been really satisfying to us that since Malprobity came online, and then since it got in Phenix, and then at the PDB, that people in general have been able to fix things and have come down close to the level in the reference structures, which are down in here somewhere. And they've leveled off now. They have to level off at some point. So to see similar things inside Phenix, again, you've been shown where the validation GUIs are with the cryo-EM one and an X-ray one, which have a lot in common, but also a number of real differences. And you get the report that Pavel took you through. And I hope that all of you will now pay attention to things like kablam. And any outlier, if you have one, is worth looking at. But at 3 to 4 angstroms, if you have no outliers, that doesn't mean it's a good structure. And that's the problem that we and a lot of other people in the field are trying to do, to find better ways to do it now. And again, you've seen some of these model to map validations. And of course, one of the great things in Phenix is when you find an outlier, you can go and look at it in COOP. Here's a plot for rotamers. And again, you can take an outlier and go and look at it. And Ramachandran plot. Now this is the one here was very badly mishandled. That's a really poor Ramachandran plot, particularly obvious in the general case here and in the Iluzale case. So the upshot of all of this is that up to about 2.5 angstroms, this kind of validation really works. If you get good scores, it means the structure is almost certainly pretty accurate. And that's just not true at 3 to 4 angstroms. So why does this happen, and what are we doing about it? So one of the basic problems by 3 angstroms is that you no longer see the nice side bumps for carbonyls, oxygens that you do at better resolutions. And so there's just no information about which direction to orient this carbonyl. And so that means that the backbone peptide orientation is pretty well undetermined. And then also side chains are very often just nubbins, and a lot of the side chain isn't visible, even if you go down pretty low in density. So there are a lot of problems. And the basic thing is that it's at 3 to 4 angstroms, and even more so at lower resolutions than 4, the map is compatible with really significantly different models. Even in this case, one's traced backwards and with lots of sequence misalignment. And this is a particularly bad case, but you see these. There was a recent structure with a whole large section, about 80 residues, where the sequence was misaligned by 15 residues. Anyway, these problems are there a lot more often than people would like to think. And what we're particularly interested in is what we think of as systematic errors. They're features of a map that are misleading even, or can be misinterpreted really easily, either by people or by programs. And so we started off doing asparagine, glutamine, and histidine flips a long time ago, and then looked particularly at backwards beta branch side chains, where if the density is sort of straight across or even not there, you can easily fit something 180 degrees backwards. And that's a doubly eclipsed bond angle at that point, bond dihedral. They're less common now that we made a big deal over them. And then again, cis non-prolines. For cis prolines, about 5% of them are cis. And so that's fine. You know, you can actually tell the difference even at 3 angstroms if you're careful about it for proline. For non-proline, you need to be closer to 2 angstroms to tell the difference. And they're extremely rare. They're only about 1 in 3,000 non-proline peptides are cis. And it went through a stage where people put in lots and lots of them. One of the, Well, anyway, one of the problems about the cis non-prolines is that they can actually, a lot of the time, if you have really bad density, a cis will fit it better because it's more compact. And so people put in a lot of them when they aren't noticing that they have them. Now we and everybody else screams about them and shows them. So this is an example of a place where there are no traditional outliers. There are some clashes, which I guess is semi-traditional, but there's a kablam outlier, this magenta broken line, and the sort of lime green trapezoid inside this peptide shows that it's a cis non-proline peptide. And also, if you are used to looking at structures, you're going to think this looks pretty weird because it's in the middle of what looks like a very good alpha helix, but this peptide is pointing the wrong way. So this doesn't have a Ramachandran outlier. So what's going on? So in fact, presumably, originally this structure had lots of outliers. And when you have an outlier sort of in the middle of space, you don't really know which place in the Ramachandran plot it belongs. And it's even worse than that, because if you get a peptide turned around backwards, you will always be in the wrong part of the Ramachandran plot, because you're moving one of the Ramachandran angles by about 180 degrees, and that's going to take you to the wrong minimum. And so presumably this point was somewhere in between, maybe out here or whatever, and it got pulled in by Ramachandran restraints into the edge of beta and even way inside beta, and it really should be alpha helical. And so some of the examples that Pavel showed of extreme cases of this will actually make wildly incorrect, fairly large percentages of a structure if you apply them right at the beginning. And so this is one of the real reasons for trying very hard to be able to make as many corrections as possible right at the beginning on your first reasonably complete model. And that's still not easy to do. It's getting a little better, but it really is worth doing before you refine Ramachandran. And my advice would be, don't use Ramachandran restraints, use hydrogen bond restraints. So Pavel and I can argue about that and continue to for a while. And it'll be fine once we have better ways of correcting more of the misoriented peptides, which is a specific thing you have to correct it in order to not have the Ramachandran restraints do harm. And when we get better at that, then it won't be an issue. So we've both learned from a lot of the PDB depositions, of course, and also there's a series of it should be the EMDR, it's the EMD resource. There are a series of cryo-EM challenges. We're in the middle of another one now that's about ligands and waters, which is going to be very interesting. So we look at a lot of these to try and learn how we can do more complex multi-residue validation and to look for problems that occur in a lot of places, because then if we find a way that people can fix them, it's going to fix lots of things all at once. So we're all for systematic errors or common problems. So for instance, one of them is that if you have an ion site that you don't know about, then the chain tracing is not very reliable. So this picture has the right answer. That's a zinc site down there. And the chain should go around it and it should have 4 cysteine ligands. But if you don't know there's a zinc there, in this case, all the cysteines tried to get into the zinc density. And even worse, in this one, the main chain went through the zinc density, which really made a mess. So that's one thing you can look for. Look for patterns that look as though the connectivity is more like all of the density that would connect into an ion. And then a misoriented peptide with the carbonyl pointing up when it should be down causes lots of problems. And kablam flags these quite well. So these magenta lines are the kablam outliers. I'll explain what they mean more a little later. But the right answer goes down, and this is then a right answer. Then, for instance, you can get kablam outliers and even cis non-pros and twisted peptides. This is a loop that's badly modeled, and you have this whole piece of density with nothing in it. And an awful lot of the model to map criteria are very sensitive to having model where there's no map, but they're not very sensitive to having map where there's no model. And so they're not great at flagging these things. But this turns out to be one end of a sequence misregister. And so this strand had to move up in order to put the leucine in here. And with it fit this way, then there are no outliers. So there are a lot of new tools to the rescue, and I've talked mainly about kablam and Sysnon Pro. Those are some of ours. I will mention some things like EM Ringer and Isolda and other things. So we'll start to explore the new tools that help you do this. So Sysnon Pro peptides are very rare, as I said. Most of the genuine ones are functionally important, and there are real genuine ones. I mean, at this resolution, there's absolutely no question this is genuine, and it is at an active site. But when you get, 5 in a row, or 12% non-pro cis and twisted, that was during this sort of epidemic of too many cis non-prolines. So at 3 to 4 angstroms, you should never fit a cis non-proline unless you have evidence from a related structure, for instance, where it's known to occur. But don't try to make all your cis prolines trans, which has happened occasionally recently. And so this is just this epidemic where people use a lot too many, and now it's back down to about what it was before. So I mentioned EM Ringer. This is now part of Phenix. And so EM Ringer is a very clever system that actually uses a survey around the position that the gamma carbon would be in, around from the beta carbon. You sort of do a circle. That's why it's called Ringer. So you make a circle around the C beta and see whether your density actually peaks at one or more of the staggered positions. But what that is actually measuring is a combination of map quality. If you're low enough resolution or really in the wrong place, you won't get peaks at all, or very, very smoothed peaks in non-staggered positions. So of map quality, and then particularly of backbone fit to the map, because what it's looking at is whether the alpha beta vector, the C alpha to C beta vector, is in the right place and pointing in the right direction. And it's a very sensitive and reliable measure of regional quality, overall quality, and it uses mostly a 20 residue window. And it's very good for finding places where your map isn't fit very well. But it doesn't show you individual residue problems. But still, local regions is very useful. And it seems to be very stable. It's the kind of thing that would be somewhat hard to restrain in refinement, which these days is an advantage for a while. People will eventually figure out how to restrain all these things, but it's a little bit like the fight between people and bacteria. So our favorite tool that is helping people fix a lot of problems is called kablam. It was invented and implemented by Christopher Williams in our lab. And what it does is use multi-residue virtual dihedrals between the C alphas. So one of these dihedrals is coming into the central residue and the second one's going out. So it's a little bit like a stretched out Ramachandran parameter. And as a lot of people have seen and used, the C alphas are certainly not perfect at low resolution, but they're the best you've got. They are the most reliable single parameters or single atomic locations of normal peptides. And then so that is what we use, for instance, to locate probable alpha helices. Again, as Pavel said, no system is perfect for that, but kablam is much better than Ramachandran or DSSP at low resolution. And then it uses the carbonyl oxygens to say what went wrong. Where do you have it fit in such a way that the carbonyl orientations are not compatible with the local C alpha trace? And the markup on a structure uses those carbonyl directions. And so for a 1% outlier, it does it in magenta, a hot pink, and for a 5% outlier, which is maybe suspicious, but not reliably bad. That means that 5% of the good data has purple outliers. But for instance, if you have a purple one next to a 1% outlier, then it probably means that the central carbonyl or the central peptide in here is what's wrong and what you need to rotate in order to fix the outlier. So here, for instance, there's a helix where the C alpha trace is just fine. It doesn't have any outliers, either traditional ones or in kablam. But if you look at the full backbone and look at the carbonyl orientations and the kablam outliers, it's terrible. And in fact, it's sort of hard to tell which direction this helix is going without knowing the numbers, because a lot of the peptides point backwards. And here it is the ones that are in the center of two adjacent kablam outliers that are wrong. And so this helix actually goes up. Then another useful thing is that if you click on one of the atoms in a kablam outlier, it will tell you how bad an outlier it is. And then it will tell you a pretty conservative probability that this might be alpha or that it might be beta structure. And it really is very conservative. And so if there's even 5% probability of helix, you should try helix. And then here in beta structure, if you fix these kablam outliers, where again, there are 3 carbonyls in a row pointing in the same direction, which is not what beta strands do. They alternate carbonyl direction, which I hope you all know. And if you fix these 2, then this turn more or less falls into place and you can get rid of its outliers. But the other thing to notice here that's almost as diagnostic as kablam, although sometimes it's not easy to fix them, is too few hydrogen bonds. So this is really a well hydrogen bonded anti-parallel beta sheet. And you can see the normal close and wide pairs of pretty good hydrogen bonds going upward from the upper strand. But between the 2 pairs below it, there's almost no beta hydrogen bonding. But this certainly looks as though it should be beta structure. And when you fix the outliers, then you get just as good hydrogen bonding in here as you did up there. So looking for regions with very few backbone hydrogen bonds is also a good way of finding local problem regions. So the idea is, if you're in the middle of secondary structure, fix it so that it's good secondary structure. And that will usually be the right answer and fix problems, other clashes and outliers of other sorts, and definitely usually get rid of the kablam outliers. And again, if you have 2 kablam outliers in a row, try rotating the middle peptide, and that also works even if it's not in secondary structures. So if we go back to the Phenix GUI, here we've got an outlier, and you can go to COOT, and this one is also actually a cis peptide. COOT marks them very similar way, but it makes them red. And COOT used to tell you when you defined a cis peptide, but if you accepted it, then it let you have it, even if it was a non-proline one. But now it will continue to flag the non-proline ones. And one thing that really pleased us in the second cryo-EM challenge is that they were looking at 3 different kinds of measures: model to map validation and pure model validation, and then CASP style validation of how close was the model to the target, which seems to us like the best thing if you have it. The point is, with a new structure, you usually don't have a target, but in the challenge, you did. And so that's a really good measure. And it turned out that kablam had the highest correlation with match to target, even more than resolution, for instance, or than any of the model to map parameters. I think the statistics are not good at all for this, so it won't necessarily hold up at this level. But anyway, it does probably have a stronger correlation. So as far as kablam goes, try and identify secondary structures early by whatever means you want to. And the most common backbone misfitting at this resolution is a misoriented peptide, and that's what kablam is sensitive to. So use kablam outliers to try to fix up the ones that you can. It's harder to do in loops because any one loop conformation is pretty rare because there's so much variety in the loops. And so kablam gets a lot of false positives in loops, and it's harder to know what to do with them. But if you can at least fix up the things in secondary structure or with adjacent kablams, you'll fix a number of the problems. And then at that point, you can start putting restraints on them to keep them good. So just to mention some things which are now becoming more and more relevant in cryo-EM as the resolutions are getting better, is things like ligand binding and ions and waters. And that's what this current cryo-EM challenge is going to be evaluating. And we've pulled up one of our old tools. I guess it's a little bit like Pavel pulling up the Rama-Z score from a long time ago. We've always had a probe score in Mulprobity, but hadn't used it very much, except in doing our own amide and histidine flips. But it's a way of taking, for instance, a single ligand, as here, and doing all atom contact dots between it and everything else. So you're seeing, for instance, it's a rough measure of how tight and good the binding is of that ligand. If there were no good contacts, it wouldn't bind there. And so if you model it and it doesn't have good contacts, either a lot of clashes or very little other contacts, then you've got the model wrong of the ligand. So here's another example. This is a spermidine, which is a very difficult ligand fitting example. Spermidine has 9 NH3s at the ends, and it has a single NH2 near the middle, but it's not at the middle. And so you could fit it with the nitrogen here or with the nitrogen here by flipping the whole molecule over backwards. And so this is showing one version of a good spermidine fit in a protein that is actually a spermidine binding protein. And it does a lot of it with van der Waals contact, actually. There are pairs of aromatics here that hold it. And this one gets a probe score of 37. The absolute numbers don't mean a whole lot, but with the same ligand, they do. But probe scores get bigger as the ligand gets bigger. And we haven't yet found a way that actually normalizes between ligands. But if you're looking at a certain one or multiple possible models, for instance, if you turn the spermidine upside down, it will then get a worse probe score. And just to show that, this one doesn't have van der Waals shown in order to really emphasize the H bonds and the clashes. So this was in a wonderful recent cryo-EM structure of the E. coli ribosome at 2 angstrom resolution. And most of the structure is gorgeous, but spermidines are hard. And so they got a bunch of them wrong, and they aren't that important for the structure. They're not the thing people look at the most, but we're interested in them as a test of how well you can fit difficult ligands. And so here, there's this huge clash at this atom, and there's nothing hydrogen bonding to the N6 nitrogen. And so if you turn it over and put N10 where N1 was and 1N where N10 was, then you shift the N6 over by one atom, and now it makes a great pair of hydrogen bonds to a phosphate and gets a way better score. And another example from that ribosome that is correct, but it can be interpreted wrong. Unfortunately, a lot of libraries make ligands neutral, and the PDB does this in its dictionaries at present, and you have to fix some of the things. It does amino acids correctly, and it does single molecule ions correctly, but it puts extra hydrogens on all the phosphates and nucleic acids. Nobody pays much attention to that one. People mostly know that that there aren't the hydrogens there. Sorry, hydrogens. I think I said it wrong. To make them neutral. And for instance, aminoglycoside antibiotics are neutral in the dictionaries, and spermidine is neutral. And so when you look at one of the rings in this antibiotic with neutral nitrogens, it's not great. If you then go and put proper NH3s in on the right places, then you get really great hydrogen bonding. And this one is sort of fun. Well, let's see, that's ring 2. I can't see my whole picture. Anyway, there's one of these rings that then has a hydrogen bond to another ring. So it's part of what helps hold the 3-ring conformation of the paromomycin. So another new tool that's relevant for high resolution is Undowser. It looks and finds all of your waters that have clashes, and there are an incredible number of different things that can be wrong. It may be the water that's wrong, but it may be what it's clashing with that's wrong, or even something more complicated. But for instance, some of the worst cases are where a water gets modeled early on in what should be an atom of the macromolecule, and that creates huge problems. and refinement unless it reassigns the waters. And even if it reassigns them without moving the model, it won't get it right. So anyway, Undowser is good for that. It's also good for finding waters that should be ions, such as this one here, that has perfect octahedral coordination around it at the right distances for magnesium. At the moment, it can't find ions that should be waters, although you can sort of kludge it by trying a water at each of the ions. Then another tool that's closely connected with Phenix is Isolde from Tristan Kroll in Randy Reed's lab, and very closely connected to Chimera X. He's finally made a tool that does what Dave and I had wanted to do for many decades. It's interactive molecular dynamics that's user guided. You can tug on things or ask it to make certain changes, like you can ask it to move along in what you think is a sequence misalignment. And it has all sorts of validation markup that changes in real time as the MD is working. And this can do marvels for fixing up 3 to 4 angstrom structures, at least marvels when Tristan does it, and pretty good marvels when other people do. So for instance, one of the examples in one of the SARS structures that we worked on collaboratively is a place where what should be a cis-proline had been fit as trans. And this meant that the tryptophan right next to it could not be fit into what is this obvious tryptophan density. And so they actually truncated it at the C beta. And if you change it to cis, then it just slides right into that gorgeous density. So if you're having real problems with your structure, you might want to learn how to use Isolde. And so these fix-ups of the SARS structures were very satisfying. They used things like kablam outliers. Here's one at a tight turn that's a double kablam outlier. And again, it's this middle carbonyl that needs to be moved. And at that point, you get way more hydrogen bonding, including some to the neighboring chain, and you take away the outliers. And then some changes in RNA where this just didn't fit the density very well. This is actually, this one's backwards. It's a syn purine when it should be anti. I mean, not that they're not sometimes syn, but clearly this one shouldn't be. And then they have these, we call bang bangs, our backbone conformers for RNA are number letter code, and exclamation points mean that it's an outlier. And when it's rebuilt, you get named conformers, and this is really a pretty good A form. And this didn't have the arms holding, this is the polymerase that copies the genome, and this is a small section of the product, and it doesn't have the things propping it up, and so it gets disordered toward the end. But this first part is very good A form. And the inhibitor, the remdesivir inhibitor, is sort of down here. So it probably doesn't affect that inhibitor, but it's pretty close to it. And then that same structure had a sequence misalignment where you can sort of see there's not enough model in this one or this one, and there's a little too much model where this tyrosine is. And when the sequence is lined up correctly, then each side chain really fits its density. This is at about 2.5 angstroms. So the punchline, one of them, is that at least one person should look at the map. This backwards tryptophan is an old classic from along, yeah, they happen occasionally, but it is a classic example of what you shouldn't have. And the worst outliers. This is a huge C beta deviation and lots of problems. But again, the goal is to have only a few outliers, not zero. And this side chain glutamine has a completely eclipsed chi angle, but it has enough hydrogen bonds to compensate for that. An eclipsed chi angle is maybe 3 kcals or so, and certainly you can compensate for it by enough hydrogen bonding. And so the real point is that you shouldn't have outliers when there's no reason for them, when there's nothing holding them in place, and or when there's a real need, a functional need for them. And so you want to look at all the problems and try to correct as many of them as you can, and then see if you can find some that are genuine outliers. They're still outliers, but they can be real. And that's true in good data. And, you know, that's the right level to come out with a few outliers, even at high resolution. And this is our group. And of course, we get all sorts of help from all the different Phenix groups. So that's it.
Fantastic. Thank you, Jane, for a great presentation. Let's open the floor to questions here. If anybody wants to stick up a hand, of course, you can talk. There's a question in the chat here. Thanks for a great talk, Jane. I wonder what could be the physical basis behind the few outlier clashes typically found in an accurate structure, given that two non-bonded atoms can't overlap.
That's an interesting, important question. They will always be pretty small, the real ones. And I think the main thing to say about it is that none of these systems are perfect. As I said, we work very hard to get good positions for the hydrogens and good van der Waals radii. And we picked 0.4 angstroms as the cutoff because a lot of the things, when we were mainly working around 2 angstrom resolution, a lot of the clashes at 0.4 were actual problems. But that's a little bit tight. And for instance, when we were working with the CAS models, we used 0.5 angstrom cutoff, and perhaps the cutoff is really somewhere in between, but it's somewhat arbitrary. And then there are very small effects like CHO-H bonds, which are real, but only at a very small level. They really are not anywhere near normal H bonds, but they can overlap a little bit, 1 or 2/10 of an angstrom. And if you add that onto the other uncertainties, you can get small clashes. But we did better without them, whereas we needed to put in things like NH's hydrogen bonding to the faces of aromatics, because we didn't get the right answer for amide flips without doing that. Also, sometimes there's a little bit of alternate conformations that you don't see very well, but that push things off a little bit. So, you know, that's guesswork, basically. If I had figured out what was wrong with them, I would have tried to fix them. But there aren't always any clashes in a good structure, but there may be some.
Yes. Great. There's, and I'm not sure if there's a validation question or a refinement question, really. Sometimes the ligand geometry is out and it's very difficult to fix. Is there a way for a ligand to fix it easily and with few refinement runs?
I think that one's for Nigel. You can fix some things maybe a little bit by looking at the contacts with the probe score. But much more general are Nigel's tools.
Yeah, it can often be lots of different things. It can be the chemistry that was used to create the restraints is not actually appropriate. It can be that there's a flip. It might be a chiral flip, or it might be just a dihedral flip. Things are very, very complicated with ligands, and if you're seeing a clash, you've got to try and think about the chemistry and whether it's a good or a bad thing, and what you can change in the general conformation of the ligand.
And it's also necessary to think about how you place the ligand. If it's low resolution, it's possible it's in the wrong orientation, and that could lead to the bad contacts that Jane was talking about, and that could be distorting the chemistry of the ligand. So you need to assess whether it's actually placed correctly as well.
Like one of the other things the spermidines do is they can be modeled off by one because they're sort of just a tube.
Mm-hmm.
And that produces bad geometry usually because you have the wrong contacts.
Yeah. This is a question for Pavel or Nigel, I guess. Is there any functionality in Phenix equivalent to the ProSmart restraints in CCP4? And I need to be reminded what those are. I should remember, but I think that's the idea of using reference model restraints, but I could be wrong.
Yeah, I think you're right, Paul. It's related to reference model restraints, in which case I don't think we have that functionality.
I mean, we do have the idea of reference model
Yes,
as restraints, but it's not done in the same way.
Exactly, yes.
Yeah, so it's I think ProSmart, that's done by distance between atoms. It's done in torsion space in Phenix. The end result is meant to be similar, but it is technically done differently.
Yes, the other difference is in Phenix, you have to define those restraints yourself manually, while ProSmart can go off to online web and fetch models from PDB and do search based on similarity, so it does more automation.
Yes, that's true. You have to. We make you provide the model that you want to do the restraining against.
But the torsion restraints do have some real advantages, so it goes both ways.
Yes, it's certainly a much nicer space to think about structures in.
So it's very easy, for instance, to do rotamer restraints and to let them loose when they need to be is very easy to do in torsion space.
Yes. And to automate and to modify the model, for example, to fix rotamers that aren't consistent between the reference and the model. That's an easy thing to change in torsion space. It's a little more challenging in distance space. Okay. There are other questions? Not seeing any right now. Well, I did want to take this chance to thank Pavel and Nigel and Jane for such great talks. So thank you so much, all of you. And thanks for all of you for attending. I saw we were up to, I think it looked like 90 odd people. So we had a good turnout, and I realize everybody's in different time zones. This is not an easy thing to pull up, but I think this works pretty well. There is another question in here. What format is the best input file for creating a CIF file for a small molecule?
So I can assume you're talking about a restraints file. And the best format for that is to know a 3-letter code from the PDB. If you don't know a 3-letter code from the PDB, the next best is something like a mol2 file or mol3D file. Those are very handy and have lots of information in there for you.
Wow.
And yeah, and so the question
and
is,
Smiles is good, but it has limitations, and because it doesn't have the 3-dimensional geometry, you can get a better representation. In Elbow, it makes an effort to get a 3-dimensional geometry, but it's not perfect. Don't use PDB. It's almost never good. And then Mol2 is even better. Yeah. So yeah, in drug discovery, structure-based drug design, most companies have other tools that will create a Mol2 file, and I would recommend that if you're doing structure-based drug design. But yeah, you can go with Smiles if that's what you really need to go with. Yes.
Yeah, I think there are some. I've certainly personally used the zinc. There's a whole set of compound information. Zinc, it's out of UCSF, and they do have ways to provide you, say, a Mol3 file, even certainly Mol2 files, but Mol3 files as well. And so that's a nice resource to use. It's manual. It's a thing on the web you have to interact with, but if you're just doing a compound here and there, that's a nice way to get something reasonable as a starting point. But as Nigel made clear in his talk, you really have to check all of these things, right?
Yeah, especially if you're doing, SSDG, structure-based drug design, you know a lot more. So you could start with Smiles and then interact with it, but Mol2 is really the best way to go.